I think as a measure of success, at least based upon the pre-registration numbers, we are going to set an all-time attendance record for this particular meeting. So that's really exciting. I know I speak for all of the planning group and the staff alike when I say that we're really excited about this year's far-ranging program of invited and submitted science that addresses many areas of pediatric investigation. I'm sure that the topics presented will provide and stimulate a lot of discussion and hopefully provide new, fresh ideas for the coming years. Although there's a whole lot of things going on at this meeting, and a lot of people tell me too many things going on, but I think there's a few things that you might want to pay attention to, and one is the plenary sessions of the sponsoring organization. In addition, the PAS serves as a host of events related to the March of Dimes Prize in Developmental Biology. This prize was founded in honor of the contributions of Jonas Salk in the elimination of polio, and recognizes a scientist who has made sentinel contributions in basic biologic mechanisms and technologies that have led to transformative changes having clinical application. About 6 of these prize winners have gone on to win the Nobel Prize. This year's recipient is Professor Rudolf Jenich of the Whitehead Institute of Biomedical Research at MIT. Dr. Janich's early work involved transgenic models during which he gained appreciation of the epigenetic influences and, as a result, facilitated subsequent creation of the now widely used induced pluripotential stem cells. This is the 20th anniversary of this award, and I encourage you to attend the session, which is on a Monday at 1 o'clock. I also encourage you to introduce yourself to a new colleague, to introduce your young faculty and colleagues and fellows to one of your old colleague friends. Please spend some time going to the poster sessions, spend a little time with, especially with the young investigators, encouraging them, welcoming them to the meeting. I hope you have a good time in San Diego, get to see some of the sites, visit with some friends, have some good meals in this beautiful city. Thank you. Donna?
Good afternoon, everybody. It's a pleasure to introduce the Joseph W. St. Jem Jr. Leadership Award, presented by the Federation of Pediatric Organizations on behalf of 7 member organizations. We are delighted that Monica St. Jem, Dr. St. Jem's wife, and Joe St. Jem III, Distinguished Professor of Pediatrics at the University of Pennsylvania, are here with us today. This award honors Dr. Joe St. Jem Jr., who is an extraordinary leader in American pediatrics. A graduate of Stanford University and medical school, he distinguished himself not only as a scholar, but also as an athlete in football and track. He trained in pediatrics, neurology, metabolism, endocrinology at the University of Minnesota, and began his career in the departments of pediatrics, microbiology, and laboratory medicine, where he developed the tools for the career that distinguished him as a physician, scientist, and educator. In 1965, he moved to UCLA and became chief of pediatrics at Harbor-UCLA. And the following year, at the age of 35, he was promoted to full professor and became chair of the Department of Peds at Harbor-UCLA. During 19 years at UCLA, he built an outstanding department and assumed increasing leadership roles throughout the university and nationally. Dr. St. Jem served in leadership roles for virtually every major organization of academic pediatrics, including president of the American Board of Pediatrics, president of the Association of Medical School Pediatric Department Chairs, president of Western Society for Pediatric Research, president of the Society for Pediatric Research, and member of the American Board of Medical Specialties, Residency Review Committee, and National Board of Medical Examiners. In 1985, he became Dean of the School of Medicine at the University of Colorado, a position he held until his untimely death in 1986. He was president-elect of the American Pediatric Society at the time of his death. Dr. St. Jem was one of those rare individuals who had remarkable clinical skills. He was a superb and beloved teacher and mentor, received numerous teaching awards, and was also an outstanding researcher who made important contributions to virology and infectious diseases. In addition, he is also remembered for his extraordinary and thoughtful leadership skills. The actual reason for this award. This award was established soon after his death to honor an individual whose contributions to pediatric are significant and broad enough to be considered as creating the future of pediatrics. It's now my pleasure to introduce Dr. Robert Vinci, who will introduce our distinguished awardee, Dr. Barry Zuckerman. Bob Vinci is the Joel and Barbara Albert Professor of Pediatrics and Chair of the Department of Pediatrics at Boston University School of Medicine. He's also Chief of Peds at Boston Medical Center. He serves as the program director of their combined residency in pediatrics, a position he's held since its inception. He's been involved in residency education for over 25 years. In addition to his work as a program director, he's been involved in fellowship training in pediatric emergency medicine at BCH. And in addition to all that, he, of course, is also an outstanding teacher, has been awarded the Jerome Klein Teaching Award at BMC, Boston University School of Medicine, twice. He's a member of the Association of Pediatric Program Directors, American Academy of Pediatrics, and of course the APS. Dr. Vinci.
Good evening, everybody. It's to the St. Jem family, the Federation of Pediatric Organizations, leadership of the PAS, and to this entire group of colleagues. I am honored to introduce my friend Barry Zuckerman as the recipient of the 2015 Joseph W. St. Jem Jr. Leadership Award. I want to first acknowledge Barry's family, his wife, Dr. Pam Zuckerman, who's here with him today, and of course his two children, Jake and Catherine. So Barry and I have been colleagues at Boston Medical Center, the former Boston City Hospital, for over 35 years. And although we take great pride in what our department has accomplished, today is the day to recognize Barry for his contributions and to congratulate him as he joins the impressive list of previous St. Jem Leadership Award winners, many of whom are in the audience here today. As mentioned by Dr. Ferrero, the St. Jem Leadership Award honors an individual who has had a major impact on child health. More importantly, the award recognizes those who've created a future. Over the next few minutes, I would like to share with you the future that Barry has created for children everywhere. Now, during most award ceremonies, it's customary to provide some important background data on the award recipient. So in keeping with that tradition, I will tell you that Barry was a graduate of Rutgers University and and completed his medical studies at the Georgetown University School of Medicine. In 1972, Barry came to Boston City Hospital with his now wife, Pam. They couldn't get into California, but that's okay. The hippies were coming to Boston, where he began his pediatric training under Joel Alpert, another pioneer in pediatrics. Subsequent training in child development with T. Barry Brazelton at Boston Children's Hospital provided the foundation for his impressive career. Barry returned back to Boston City in 1977, and during his tenure in our department, he served as the division chief of developmental and behavioral pediatrics, and for 19 years as the chair and Alpert professor at the Boston University School of Medicine. However, there's more to Barry, and in the spirit of the PAS, I've been provided with a few slides to tell the rest of the story. Early in life, it was clear he was destined to study child development, here demonstrating the social smile. He helped us understand the challenges of toilet training. We learned the importance of healthy nutrition and eating with your hands, and the importance of developing social skills while playing with your friends. And might I say, demonstrating the straightest part in the hair that I've ever seen in a child. Barry's most widely known accomplishment is Reach Out and Read, a program that promotes literacy and school readiness by utilizing pediatricians to provide books to parents and children during well child visits. Here is Barry practicing Reach Out and Read. Although there is more to tell about his early years, I must fast forward to his medical career to discuss his ideas, his creativity, and his vision. So how did Reach Out and Read start? In the late 1980s, Barry and Robert Needleman, who was then a trainee with Barry, noted how parents gravitated towards books in the waiting room of our ambulatory clinic. The gratitude of families sparked Barry and his colleagues to place even more books there, and soon they were gone, highly desired by parents of all backgrounds. That simple observation led Barry to start Reach Out and Read in 1989, first at Boston Medical Center, then local community health centers, and eventually national dissemination. Having just celebrated its 25th anniversary, Reach Out and Read, now with over 5,200 sites, distributes 6.5 million books per year while serving over more than 4 million children. Okay, so he convinced an entire nation to read to children. What else could he do? Well, Reach Out and Read is just one example of Barry thinking outside the box. In his well-cited research of studying the long-term neurocognitive outcomes of children exposed to cocaine, Barry understood and subsequently emphasized the far greater importance of exposure to alcohol, tobacco, and the quality of the environment, well before many others were aware of these challenges for children. Barry was not content to just understand these challenges, but he wanted to solve them and made that his career focus. In 1993, Barry recognized that child health problems often arise from situations that violate existing laws and regulations. Like many pediatricians, he was frustrated by this, but struggled for a solution. Once, when expressing his concern about the plight of children living in poverty, a lawyer colleague asked Barry, "What does the law say about these health-related issues?" Barry got that look in his eye, came to work the next day and said, "We're hiring a lawyer." And he recruited a lawyer to become a member of our department. That simple idea grew into the medical legal partnership. The MLP program now provides legal assistance to parents and families, education and training for healthcare staff, and advocacy for health issues. MLP has since disseminated to 260 sites across the country. I know Barry is proud of the national dissemination of MLP, but to be honest, what he really loved was having our attorneys prod landlords, secure government benefits for families, and fight with Medicaid, insurance companies, schools, and other bureaucracies. Barry never liked rules and always fought for those families who could not fight for themselves. He was also open to any idea. One day, a college student read a story about Barry. She wrote to him and suggested that students could play an important role in healthcare by assisting families with making health connections in the community. How many of us get those notes from folks who just want to come in and discuss ideas? So what happened to that conversation? Well, I can tell you from that conversation, Health Leads, a program that created by Barry and the college student by the name of Rebecca Oney. And last year, Health Leads connected thousands of families to community resources. Barry also played the lead role in the development of Healthy Steps and worked alongside Ted Kennedy on a number of health policy challenges. Basically, Barry has conceived, implemented, and disseminated program after program to improve the health of our nation's children, programs that are as effective as they are innovative. Barry's creative thinking and innovation in our department led colleagues to develop comprehensive programs for the care of children with chronic disease, most notably patients with sickle cell disease, as well as programs to provide a comprehensive approach to children with special health care needs. Barry has guided his colleagues to develop programs that push the boundaries of traditional health care. A New York Times reporter, in describing Barry's career, once wrote that Barry, quote, deserves his own wing in medicine's Hall of Innovation. I'm not sure we have such a Hall of Innovation, but if we did, naming it after Barry would be a good place to start. However, what we do have is an award that honors a pediatrician who is a role model for others to emulate. Barry Zuckerman has been an outstanding leader, innovator, educator, and advocate. It is my honor to introduce Barry Zuckerman as the recipient of the 2015 Joseph W. St. Jem Jr. Leadership Award. Barry Zuckerman.
Thank you. Thank you. Okay, let me see if I can do this. Thank you, Bob, for that lovely, warm, and personal introduction. And I particularly thank you for going light on me. For those of you who know Bob, he has a tradition of roasting our chief residents at the beginning of the year. And I, again, appreciate the kind words without the particular roasts. It is a special honor to be included in a group of illustrious colleagues who are past recipients of the Joseph St. Jem Jr. Award. Interestingly enough, one of those recipients is my cousin Lou Barness, actually my father's cousin, who won it a number of years ago. So, at least in my family, it was pretty significant, this kind of award, and anything that Lewis did, who was considered a hero and idol, is just great. The real honor and privilege for all of us is caring and making a difference for children and families. Our best work is catalyzed and shaped by observations, stimulating discussions, and sharing strategies, ideas, and passion with our colleagues. I am fortunate to have many special colleagues at BMC and nationally, whose ideas, feedback, and collaboration has been and remains the cornerstone for everything I do and have accomplished. While there are too many colleagues to name, I want to take this opportunity to thank you with great gratitude and appreciation. I do, however, want to mention by name a few. I have been fortunate to have two talented and passionate mentors, Barry Brazelton and Joel Alpert. Barry will be 97 in a couple of weeks, and Joel unfortunately died a little over a year ago. Both were wonderful and model pediatricians who both changed pediatrics. Joel by advocating the need for primary care training, which emphasized continuity of care as a cornerstone of effective care. Barry helped all of us, doctors, nurses, researchers, and parents, see infants beyond reflexes as responsive in learning, and also taught us how to use an infant's behavior to form a special relationship with parents. Both were models of a lifelong and enthusiastic commitment to children and families. Barry and Joel also believed in me and my work. Just as a healthy attachment to a parent is necessary for a child to feel safe to go out and explore the world, they provided me with a secure base to explore. I am deeply grateful to both of them. Next, I want to mention a few special colleagues at BMC. First, Steve Parker, who died too young. Steve and I had great discussions, sometimes disagreements, but his views always sharpened my thinking. Steve, I miss you. Bob Vinci, Howard Bachner, and I, three boys from New Jersey, provided complementary and overlapping skills and knowledge and deep commitment to the pediatric department at Boston City Hospital, now Boston Medical Center. My accomplishments are in a significant way due to them in so many big and small ways. One other name I want to mention is my wonderful assistant for many years, Missy Brennan. For any of you who are fortunate enough to have Missy help you with a variety of problems, you know how terrific she is. Missy, I thank you for the great care you have taken for me and for your insights and humor. Finally, I have been fortunate to work with and mentor many younger colleagues, fellows, and residents. This continues to provide me with great satisfaction and challenge. Many are now making significant academic and leadership contributions, and all are passionate about making a difference for children in their families in the U.S. and globally, for individuals and for populations. The choice of our work is informed by our identity, how we see ourselves. For me, my work has been shaped and directed by many experiences on a foundation of values learned from my religion and my family. My father went to college for 8 years at night to get his Bachelor of Science while working during the day. As a biochemist, he took great pride and satisfaction in helping to produce Rogam, which has saved literally hundreds and thousands of babies around the world. My mother's love for me was endless, and she also set high standards and exceptions and expectations for me. My grandparents were immigrants and not always treated well with respect by the healthcare system. My brother, who is 66, has a significant intellectual disability. When we were growing up, there were no educational opportunities for him beyond 2 years of kindergarten and few health services available to him and my family. The best pediatricians of the day recommended that he be placed in an institution at age 7 for the overall good of our family. We made the 6-hour round trip monthly to visit, and it wasn't until his late 20s when we were able to place him in a newly developed community setting. His life and experiences and the local advocacy by my parents has been an inspiration and driver in my career. As a child of the 60s, my efforts expanded beyond children with disabilities to include all children, especially those whose race, income, or immigration status were a barrier to health or education. Given what Robert Putnam documented in his new book about the diminished social mobility in the U.S., we still have much to do to help low-income children. Finally, I am the husband of Pam, who, as my children would say, is the real doctor in the family. Her experiences, insights, and clinical expertise are a significant part of my understanding of clinical pediatrics. She is truly a master clinician. More importantly, however, is her love and friendship. Our children, Jake and Catherine, are the most important part of our lives, and and where we learned that there are no boundaries or age limits on caring about your children. It is also where our understanding of ourselves was shaped by the normal challenges of being a parent. My family, both by family of origin and by marriage, is at the heart of my identity and motivation. I've always wanted to give a TED Talk because of its emphasis on ideas and challenging conventional practice or wisdom. So here goes. And first, okay. The accomplishments and pace of biomedical innovation has been spectacular. Death rates from infectious disease, cancer, and most conditions have drastically fallen, but by every health, societal, social, and educational metric, low-income children are doing less well than their well-to-do peers. We must challenge the structures that lead to poor health and the loss of potential, even when they are our own structures and traditions. Not only do we need new ideas, but the strength and fortitude to be willing to change our habits. That is what we ask parents of our patients to do when evidence suggests such changes will improve their health. Let me suggest one change based on my academic work over 30 years ago about the importance of maternal health to children. These individual items got my attention mostly through clinical work, and much of my early work was not doing research, but wrote some reviews and commentaries, although I did do some research. The factors of maternal health that got my attention were maternal depression, drug, alcohol, and cigarette use during pregnancy, trauma to both children and parents, particularly mothers, and preventing unplanned pregnancy. Since those observations, the data has accumulated significantly, showing the significant impact on all children, both normal children and children with chronic health problems. These problems accumulate and affect children's outcome. But even more important than that is the fact that these problems, besides documenting the impact, there are evidence-based interventions to address them. And that evidence-based intervention, coupled with my observation of practice over the years, is the best way to help children is to help their parents. The best way to reach parents is through their children. That becomes a key role we have in some ways differently than anyone else. And I think the implications of that is that we need to reframe our work to a two-generation approach to pediatric care. Many clinicians now are already doing this with one or the other, but we need to screen and address all four of these maternal health problems because of their impact. We also need to not only identify these problems, but to develop a system so we can refer parents to get the evidence-based treatment that they need. And those systems need to ensure that parents don't fall in the gap that goes through with this. I want to make one particular mention of one of those, preventing unplanned pregnancy, because the other three are more common now. When you have an intervention that's close to, as LARCs, long-acting reversible contraceptives are, that are 99.5% effective and safe, it seems to me it's incumbent upon us ensuring access to parents. It's great that we have women's health clinics and family planning clinics, but it's us as pediatricians who see young mothers regularly. What is our role? Minimally, our role is to ask within the first few months of a child's birth, when do you want to have your next baby? We've had some colleagues started to do this, and the parents have been extraordinarily receptive of addressing this question, and it allows us to work with our family planning group to get a quick referral. I also have a fantasy that maybe someday some pediatricians, not just adolescent pediatricians, will put in implantables in the arm. This is a 5-minute procedure. Now, some of you are going to say, how are we going to have time to do that? Unlike other things that myself and others may ask, the RVUs for doing that are more than primary care visit, and you can bill both. The laws about contraception and family planning allows you to bill for it. So if you would do that in your practice, you will be able to more than double your visit. So therefore, it's doing good and actually getting paid for it. But even if you don't do that, think about the framing, the orientation of a two-generation model of health, and will that get us to where we want to go? I think it's important. It moves us in a very real way to follow the evidence. And I would like you all to think about that in a complete way. I know the response is we're already doing so much, but as I said before, change will take some strength and fortitude, and we have to change our habits. Next, the second part of my talk, I want to demystify the idea of innovation, and particularly the notion of clinician-driven innovation. Insurers, hospital administrators are all for innovation, and the primary emphasis is efficiency, saving cost, certainly, reducing errors, but that's where they're coming from. Who are the people who are going to look at innovation to meet the needs of patients, fill in some of the gaps in their care? It's primarily us. And the notion of what I'm suggesting to you is this sequence here. It first starts with observations and experience. Just as Bob pointed out, when children weren't doing well when they entered school, our patients, and weren't reading, and then we found out that they didn't have books in the home, then we gave it to them. When children's asthma was being interfered by poor housing conditions, and we would call up and get nowhere with the landlords, we got lawyers. So the first thing is, when you see a problem, and more than one patient has it, it starts to be a theme. Instead of complaining about it, is there an answer? Not always, but maybe you can think about it. So then you have an idea what to do. And this MVP means minimally viable product or first draft. So if you have an idea, whether it's a book or a lawyer or any other idea you have, you do it with 5 or 10 patients as part of your clinical care and see how it goes. You may not need human studies approval, depending on the level of risk, and it's only 5 or 10 patients. But there's an iterative process with these ideas. You do it 5 or 10 patients, and maybe it really works well for 1 or 2 and not for the others. You make a little adjustment, you do another 5 or 10, you call a colleague at another institution, they start trying it, give you some feedback, and before you know it, in 6 months to a year, you have something that you've refined during this very quick iterative kind of process of learning. Early on, it doesn't have to be systematic with feedback. It's probably not something you're going to want to publish. Later on, you're going to want to do the feedback, the iterative process, more structured. At that point, after this small-scale iterative testing, that's when when our academic pediatrics come in, we do evaluation. We do randomized controlled trials or other things to do. What I'm trying to suggest is there are other things we can do before the randomized controlled trial. And this is particularly for those of you who aren't independent investigators. These are for people in practice who have a little bit of time and want to make a difference. You and your experiences should lead to ideas that you can test, and then at some point, either you with colleagues or someone else can evaluate it. And then along the sequence is scaling up and dissemination, and that really amounts to understanding the key aspects of it, some theory of change, because this is all about changing behavior. I'm talking about innovation to change behaviors, not to create a new pill or anything like that. Other people do that, not a new technology. We're talking about changing behaviors to either help manage a disease or to keep children healthy. And then once you scale up and have something, then you move to quality improvement. You try and keep making it better. So keep this sequence in mind. Traditionally, we're at the evaluation RCT stage, but there's other things that some of you can contribute, and hopefully we'll think about it. Nope, going the wrong direction. So I want to share with you one of my new ideas that I'm thinking about, and it's the use of digital media, which is kind of ironic because I'm the least technological person one could meet. And What we do now with parents, and we do it every day with patients and children, is we explain what's going on, and sometimes we draw pictures on yellow sheets of paper or some other piece of paper. Sometimes we give a pamphlet. That's the way we communicate information. Now, I want to use an analogy of the car mechanic. When my car starts doing something, like making noises or not working well, I go to the car mechanic. He does his history. He asks me some information. He does a physical. He looks under the hood. And then he tells me what the problem is. Well, I have no idea what he's talking about. I don't know the names of the parts, and I don't know how they work together. I can't help that many of our patients feel the same way, particularly low income, particularly bilingual, particularly patients with low education. And we can do better. Just like if the car mechanic had some diagram with arrows and names, maybe even I could understand. So I want to talk about digital media for use as a tool, and really what I mean by a tool, this is not standalone. This is to be used by doctors, nurses, and others to communicate more effectively. And there are a couple of key principles that I'd like to communicate. One is we should show them what's going on and what to do, not tell them. This includes pictures, animation, video, and interactive aspects. I'm going to show you some examples. Part of what's important when you do any of these is an emotional connection. And the converse of the opposite of emotional connection is when you see some TV commercial, it's just some information. It's just some person talking at you, and the information's there, but you can't relate to it. If you want to think about emotional connection, think about how advertisers market and sell us. They work very hard to make us want to buy something or be like someone, and we make fun of it, but if we want to communicate effectively, there's something important there. The other thing is, and I've looked at a fair amount of videos, probably enough with talking head experts. Why don't we start using voices of parents, faces of parents, languages of parents, the real parents, again, particularly, I'm focusing primarily on low income, but this really can cross all boundaries. But given that low income kids at a level of income we're talking about is half the kids and they carry a burden of most of the disease, there's something to say. It should be non-scripted. This should be spontaneous. It should not sound like a commercial or an advertisement. And I think there's something for the future, what we're calling wraparound communication. We have 20-minute visits, but with this technology, we're going to communicate before visits, after visits, between visits, and what is it that we're going to communicate? How can we, in a very effective way, not just throw information at people, how can we do something better? And I want to give you a couple, there are two products, or not products, but pieces I'm working on. One is a group of disease management ebooks. We started with diabetes, we're doing something also with preventive cardiology, but this is open to everyone to do animation, videos, and interactive stuff. So I'm just going to work you through a couple of these slides. Obviously, insulin and food, and here you see the liver putting out glucose and no insulin coming from the damaged pancreas. I just want to give you these are called storybooks. So this will have, I'm doing this with WGBH Public Television in Boston. This will be colorful, this will be interactive, but I want to give you the idea. This shows you're getting your long-acting insulin, which gives you the keys, which allows the glucose to get into the body. This one is the interactive part and asks, the blood glucose, the child's going to do some activity and the level is fine. And the question is, should she eat a snack before her activity? And there's a yes or no answer. And the yes answer is she eats her snack and she goes about her activity. And on the other hand, if the answer is no, she also goes about her activity, but you see, through a visual what's going to happen to her. Ketones is our biggest challenging things. How do you explain ketones? And I've spoke to diabetes educators, it's their challenge too. So here you have a child with glucose and insulin in their body, and then you see the insulin going away, the lack of keys, you see glucose and ketones, and you see these kinds of symptoms. This is very simple, but maybe this can help us and help our patients understand what they have and really sort of manage it. I imagine these series of disease ebooks should be for almost all of our diseases, particularly the common one. We can do better explaining to people. Well-educated parents find the materials. The interesting thing about diabetes, and I'm doing this with our endocrinologist, Angelina Bernier, is I asked her, well, there must be something out there. She said, no, there's one Bible, it's very text-dense, and, that's really what's there, and that's why we went with that. I now want to, the other arm of this is for primary care. I want to use visual images to get the attention of some issue related to learning or child development to use in the office with a handheld, to be followed up, to be sending to the parent when they leave, and also with some links related to that topic. And we will also be able to measure whether they open those links or not, or whether they use it. This is just an issue of testing. But I again, not a technology person, but also not a filmer, I did these as prototypes so I can show, the real media people what I'm looking for. So I would like you just to, I want to run you through one or two.
One is I am showing my daughter the pictures here and hoping that she will repeat what I say. She's 2 years old, so she should be talking more, but she's not very verbal. But she will repeat what someone tell her to say. So if I do that, I'm hoping that she will talk more in a sense and give us some synthesis, because she doesn't do synthesis at all.
That was, clearly, and then I just the same mother. But I want you to think about who's showing this.
See the baby lamb?
Did I lose the sound? No, it's okay.
Doggy? See, these are puppies. Puppies? Yeah, puppy. Good job. Puppy?
Yeah, let her, whatever you want her to do.
Let's open it up and see what's else. Now there's 3 puppies. You see? On to the next page. Here you go. And what are those? Those are ducks? Yeah.
1, 2, 3, 4, 5, 6, 7.
Again, this is about showing. This is about the people we're talking about. This is demystifying, and hopefully it's an emotional connection. And I'm going to do one more, if I can get this on. This I pulled from YouTube.
Ma, 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 ma. Ma, 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 ma. Ma, 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 ma. Say it, London. Good girl. You're only 4 months.
Out of the mouth of babies. I want to thank you. Hopefully some of these ideas will stimulate your own ideas. I will be looking for colleagues to partner with us and participate with these kinds of things. Some of you must have technological skills and like to create things. So again, thank you all very much.
Receive your plaque.
Over there.
Over there.
Thank you, Barry.
Thank you all.
Oh, yeah. Oh, shut.
This is the one without the tie.
Oh.
Okay, thank you.
Okay. Our next event is started with much feeling. It's a great honor to be up here with Jake to introduce the inaugural Debbie Anagnostelis keynote address. I'm going to turn it over to Jake to tell you a little bit about Deb.
Thank you, Donna. Well, this is really quite an occasion today to be able to introduce the Debbie Anagnostelis keynote address. Debbie was an amazing person, and she was our director of our home office, and she was an incredible leader for our collective societies. She had an amazing impact on academic pediatrics, and we're deeply indebted to her. Debbie passed away this past March, and really we're quite heartbroken by that. She had an amazing career, and she had worked for the pediatric academic societies and the various organizations for over 35 years. She started back at the University of New Mexico when she was the secretary for the then secretary treasurer of the SPR, and she grew into these leadership roles over time, moving to Chicago and eventually moving to the Woodlands to become the executive director of the home office, which included the Society for Pediatric Research and the American Pediatric Society, as well as lots of other activities related to this meeting. Debbie had many friends and colleagues, and she took great pride in her role as an academic leader. This is a picture of her with Gail Demler around the time of the 75th anniversary of the SPR. And Dr. Demler was a past president of SPR. And you can see her over the many years contributing with her staff, enjoying her roles immensely. Really quite an amazing person. So here she is with the current director of the APS. And she was actually a Thomas Hazinski Distinguished Service Award recipient. And this is in recognition of her incredible contributions for academic pediatrics, and really very, very deserving. Well, Debbie continued to work as the executive director for years and years, up until her death in March. And she was survived by Vicki Long, her wife, but her family is also here today, and we want to recognize them. In recognition of all of Debbie's contributions, we've named the inaugural lecture after her. So this is the Debbie Anagnostelis keynote address, and we're really thrilled to be able to give it here today. I'd like to note that she has family attending, including her parents, Barbara and Gus Anagnostelis, as well as Jim and Peter Anagnostelis, that's her brother and nephew, Scott Anagnostelis, who is her brother, Mary Algier, her aunt, Shelley Algier, her cousin, and Kim Algier, her cousin. Not attending today are Vicki Long, her wife, as well as Dick and Rosemary Bloom, her great uncle and aunt. So it's a wonderful tribute to all of Debbie's incredible contributions to our societies and to our field. I'm so thrilled that we can name this keynote address in her honor. With that, I'd like to bring up Mike Folds, who's going to introduce this year's Debbie Anagnostelis keynote address.
Before we get to the keynote address, I'd first like to introduce Tina Chang, who will present a brief video on the 7 great pediatric research achievements of the past 40 years. Tina?
We live in extraordinary times, thanks to medical, public health, and scientific advances that have decreased mortality, increased life expectancy, and improved quality of life. 40 years ago, we didn't know what HIV was. Now, antiretrovirals are saving lives. Cystic fibrosis life expectancy in the United States was in childhood. Now, it's closer to 40 years. Haemophilus influenza killed thousands. After the vaccine, it's very rare. We do incredibly important work in pediatric research. However, we also know that there are threats to continued support of our work. For decades, the importance of biomedical research was unquestioned, but times have changed. Ezekiel Emanuel wrote in JAMA on the future of biomedical research that researchers have to do a better job of explaining the value of biomedical research. Frankly, we in the public at large take for granted the tremendous research discoveries that have occurred. For this reason, the American Academy of Pediatrics and its partner pediatric professional organizations have embarked on a campaign on the 7 great achievements in pediatric research of the last 40 years. Because of research in these 7 areas, children are healthier and safer today and will grow up to be healthier adults. Of course, there's many more than 7 great achievements, but we do know that we need to continue research and its translation to practice and policy, and that we are at the cusp of many more discoveries to address old and new health challenges. The campaign began with a well-attended congressional briefing in December, and today I'm pleased to introduce a video for the public and policymakers to show the value of the work that we do. We hope that you'll join us in this campaign. Tell your story. Talk up the achievements of past research and of your research. Thank your congressional representatives when you receive a federal research grant or when you have important findings. Translate that research to the public and to policymakers. Emphasize that children's health is a foundation for adult health and that we need to be investing in research early to prevent disease and invest in finding cures. So visit the website, aap.org/7achievements, and in there you will find a place where you can take action. Just with a touch of a button, you can send a letter to your congressional representatives talking about pediatric research and its importance. Or when you get a research grant or you have research findings, there also is a place with the touch of the button that you can customize a message to your congressional representatives. Relay the message that pediatric research investment really does pay off in improving quality of life and in saving lives. We hope you'll join us and enjoy this video.
My name is Sandra Hassink. I'm president of the American Academy of Pediatrics. The way pediatricians practice medicine today is made possible by decades of pediatric research. Every child needs a strong foundation to ensure a healthy life. From nutrition to supportive relationships, pediatric research has taught us what we know about what makes children thrive. Pediatricians have long understood the connection from research discovery to medical treatment. Pediatric research helps children start out healthy and stay healthy into adulthood.
As we think about the last 40 years of discoveries and innovations in pediatric medicine, 7 great achievements in research come to mind. When I was training to become a pediatrician, about 20,000 U.S. children had Haemophilus influenza or HIV infections every year, and up to 1,200 children died. Today, because of the vaccine, current doctors in training, including my interns, have never seen a case. SIDS is a leading cause of death for infants between 1 month and 1 year of age. An awareness campaign urging parents to put their babies on their backs to sleep, not their stomachs, cut the rate of SIDS in half. Acute lymphocytic leukemia is one of the most common childhood cancers. In the past, it meant certain death. Cancer research has led to the creation of medications and treatments that destroy these cancer cells. Today, more than 90% of children with this disease survive. Research showed that premature infant lungs did not have enough of a compound known as surfactant to breathe normally. In 1990, the FDA approved supplementary surfactant. As a result, more babies born prematurely go on to become healthy adults.
We were really excited when we found out we were first pregnant. I went in for my 20-week checkup. I remember the doctors telling us that I was in preterm labor. I tried to stay as positive as possible because I knew at that time really it's very rare that a baby would survive. They said I had to do a quick cesarean. I just said give him the best fighting chance possible. Kingston was 1 pound, 3.5 ounces. So, I mean, you're talking about a baby who can literally fit in your hand. I had a nurse come in at the time, and I started talking to her about all the research that had been reading on Watch Your Dogs about surfactant therapy, and the shots that kind of help develop the baby's lungs and help them be able to breathe. If it wasn't for the surfactant therapy, then honestly, we could say we don't know if Kingston would still be here.
And today,
He's a 3-year-old going on 13. He's my pride and joy.
you would never know that at one point he couldn't breathe on his own for longer than 15, 20 minutes.
20 years ago, 1 in 4 mothers with HIV transmitted the virus to their babies. Discovery of antiretroviral medications has reduced transmission by 2/3 to the point where it's very rare today. Sickle cell anemia is an inherited disorder that causes a person's red blood cells to change their shape. The cells are more easily destroyed and tend to form clots, causing extreme pain, infection, and strokes. Research led to the development of new medications, preventive care screenings, and standardized disease management.
About 40 years ago, the average life expectancy for sickle cell disease was in the teenage years. It has now increased to about 40 years. My mom was born with sickle cell disease. She passed away when she was 39, and that really affected myself and my family. I am really grateful for the time and effort that's been put into pediatric research to help extend life expectancy. I have my life because of that.
Research led to the development of car seats, seat belts, and graduated licensing programs. In infants, using car seats decreases mortality by 71%. In older children, the use of seat belts decreases mortality by about 50%. We all should be incredibly proud of the discoveries that have been made. But we still have a lot of work to do.
It's very important that we continue to make sure that we're funding pediatric research.
Funding research is really what makes it possible to advance child health.
We have made incredible discoveries, and those investments have paid off in saving lives.
Thank you very much, Tina. That was great. I strongly suspect that the topic of our keynote speakers will become the 8th greatest achievement. At this time, I am pleased to introduce the speaker for the inaugural Debbie Anagnostelis keynote address. Dr. Richard Lifton is the chair of the Department of Genetics, Sterling Professor of Genetics and Internal Medicine, Executive Director of the Yale Center for Genome Analysis, and an investigator of the Howard Hughes Medical Institute at Yale University School of Medicine. Dr. Lifton has used human genetics and genomics to identify mutations that identify key pathways underlying common diseases, including hypertension, congenital heart disease, myocardial infarction, osteoporosis, cerebral hemorrhage, and neoplasia. In the area of hypertension, these studies led to the recognition of the key roles of sodium and potassium handling and have led to new therapeutic advances to treatment and prevention. He has received the highest scientific awards of the American Heart Association, the American Society of Nephrology, the Council for High Blood Pressure Research, the American Society of Hypertension, the International Society of Hypertension, and the International Society of Nephrology. This afternoon, Dr. Lifton will discuss genes, genomes, and the future of medicine. Dr. Lifton.
Thanks very much, Dr. Folds. It's a real honor for me to be here this afternoon, particularly in the setting of this symposium. First, in the memory of a leader of this society who was taken from us far too early. And secondly, to bookend a symposium that goes from basic science all the way through to clinical care. And as a physician scientist myself, I'm well aware that the best basic science has little impact unless we ultimately deliver it to the patients who need it. So it's really thrilling for me to be speaking in this symposium with a distinguished honoree. So what I want to speak about today is how genetics and genomics is having impact on our understanding of diseases that failed to yield to traditional approaches to understanding. So when we think about what we want to do in biomedical investigation, we want to understand normal, healthy, and disease biology in the expectation that we do our best in medicine when we understand the fundamental causes of disease, and this enables development of new approaches to prevention, early diagnosis, and therapeutics. So Mendelian genetics has had profound impact on this understanding. And the importance of these single mutations with large effects are that for diseases that have been entirely mysterious in their causation, they can establish the causal relationship between genes, pathways, and human traits. And these can provide direct routes to genetic testing that clearly establishes disease pathogenesis, often ends diagnostic odysseys, and frequently suggests specific therapeutic approaches to disease. Although until we know what the exact causes are, we frequently are completely in the dark as to whether we will be able to devise effective therapies or not. And even when these are possible, frequently these can take years and decades to ultimately develop. But importantly, I want to make the point that the understanding of these rare diseases, which are seen commonly in the pediatric population, can also have profound impact not just on the patients who have these diseases themselves, but on many other patients who have common diseases that are exemplified by these extreme rare forms, and that these can lead to new therapeutic approaches. And as examples of these, I've listed a few of these. Now, of course, many of the diseases that we're talking about, the affected individuals need therapies, and these are frequently slow to arrive. But this past year, cystic fibrosis has had the first real advance in the specific treatment of the disease that shows promise with development of a small molecule called Kalydeco that is used clinically to cause the channel to attain an open state that is normally defective, and that this has had impact for a small subset of patients with cystic fibrosis, but is very promising for potential therapeutic approaches to other patients with cystic fibrosis. But there are many disorders that are unusual that have suggested specific approaches to therapeutics that are now being approached for treatment of diseases in the general population. So if you are missing a specific sodium channel called NaV1.7, you are impervious to sensory pain. So it is no surprise then that if you wanted to develop a new anti-nociceptive, that if you had a small molecule that selectively inhibited that sodium channel, you might have a drug that would be broadly applicable to the general population. And importantly, this channel is not expressed in the brain, so you would not need to have the general effects in the CNS that we get with narcotic analgesics, for example. There are many other examples along these lines. PCSK9 is a very attractive target for LDL lowering to prevent heart attacks. A small number of neurons in the brain express orexin receptors. These were discovered as being mutated in patients with narcolepsy, and this has led to a new sleep medication that hit the market in the last year, which selectively targets just the neurons that are involved in regulating sleep and wake, again, rather than the general anesthetic approaches that are commonly used. And for those of us who are getting later on in life, it's optimistic to understand that there are rare forms of Alzheimer's disease that have particularly early onset that can be caused by mutations in the amyloid precursor pathway, and that these mutations have validated the idea that if you could interfere with the development of the amyloid plaque, you would likely be able to attenuate or mitigate the effects of this and retard the development of Alzheimer's disease. So for all of these reasons, I've been passionately interested in rare mutations with large effect for the impact on the patients who suffer these disorders, but for the potentially broad application to the general population. So as mentioned, we spent many years tackling a complex problem in the adult population of hypertension. It affects 1 billion people worldwide, and when I started in this area, it was passionately debated as to whether hypertension was a primary disease of the brain, heart, kidney, adrenal gland, or the vasculature itself, and this obviously thwarts approaches to rational therapy. So we scoured the world looking for children with early onset of the severest high and the most extreme low blood pressure that was compatible with life, and identified which of those were segregating as simple single gene traits that we could use the developing tools of the Human Genome Project to map and ultimately identify the underlying disease genes. And we demonstrated that these mutations are not distributed across many organ systems or the physiologic landscape, but instead converge on a single final common pathway that regulates how the kidney handles salt. And mutations that increase salt reabsorption by the kidney increase blood pressure, and mutations that lower salt reabsorption by the kidney reduce blood pressure. And while there are diverse effects on potassium, calcium, and magnesium, if you know the vector of what's happening to sodium and chloride, you know what is happening to blood pressure. And indeed, it is telling that for 5 of these genes, loss of function mutations drive blood pressure to the lowest levels compatible with survival, and gain of function mutations drive blood pressure to the opposite extreme, telling you the power of this simple mechanism to drive blood pressure across the entire spectrum seen in the human population. And we now can specifically treat many of these disorders with simple therapies that are geared to the underlying abnormalities. But in addition to treating these specific rare disorders in patients who have them, this has had impact on the way we think about hypertension in the general population. And so despite the admonitions of the Salt Institute, who have long since learned that if they want to sell more beer, the best thing to do is to get you to eat salty food, we now recognize that lowering salt balance is a key goal of reducing hypertension in the human population. And the World Health Organization, NIH, the Heart Association, all agree with this, and more than 30 countries worldwide have initiated programs to reduce morbidity and mortality from cardiovascular disease by reducing dietary salt. And the Institute of Medicine has released a proposal in the U.S. Of how to achieve about a 25% reduction by focusing on processed and fast food restaurants. So what I want to talk most about in this discussion this afternoon is how the change in the cost of DNA sequencing is leading to another revolution in human genetics and genomics. So when we started the sequence of the Human Genome Project in 1998, it cost about $100,000 to sequence 1 million bases of DNA. This year, that same 1 million bases of DNA can be sequenced for about 2 cents. So this is nearly a 7 order of magnitude reduction in the cost of DNA sequencing. And recognizing that this was coming back in 2008, my colleagues and I decided that while we would love to be sequencing whole genomes, that was still going to be pretty expensive for a while. And it's still a little more expensive than we can routinely use. But we thought, boy, there's an awful lot that we can do outside the context of sequencing whole genomes, and we ought to be able to do it very inexpensively. And specifically, there are about 21,000 protein coding genes in the human genome, and we know what happens when about 3,000 of those genes are mutated in the context of a human being. And as a consequence, not to put too fine a point on it, when my friend David Valley is asked what remains to be done in Mendelian genetics, his answer, which I think is apt, is practically everything. The vast majority of genes, we don't know what happens when they're mutated in humans, and this gives us a relatively large terrain to be thinking about. So this inspired us to develop methods for rapidly and efficiently sequencing just the protein coding parts of the genome, because that part of the genome comprises only about 1% of the human genome. And as a consequence, even while it was very expensive and still is quite expensive to sequence whole genomes, and we're optimistic about getting to a stage where that won't be such an issue, but it still is today, we thought we could get a march on this problem by devising methods to rapidly and efficiently sequence just the protein coding parts of the genome, and we've developed this technology in collaboration with a company then called Nimblegen, and we reduced it to practice and have been relentless in driving down the cost of this to increase the number of experiments that we could do, and this summer will be down to about $350 to sequence all the genes in the genome for a research study. So we can do a lot of experiments with this, and the rest of the field has figured this out as well. So we performed the first reduction to practice and demonstration of a biological outcome from such a study by making a clinical diagnosis in 2009 by using this approach. And Jay Shenduri and colleagues at University of Washington at about the same time did a similar approach which led to the first diagnosis of identification of a new disease gene by this same technology. And from this standing start in 2009, this has become used worldwide, and in the last year there are about 1,600 papers published in the biomedical literature using this technology with a goal of trying to flesh out what the remaining genes in the genome are doing in the context of a human being. NIH recognized that this was a good thing to do as well, and they set up centers for Mendelian genomics, and there are 3 national centers whose remit is to identify and define the causes of all human diseases that are caused by mutations in single genes. And we have recruited patients from all over the world. No surprise, many of these are from the pediatric population, and this has been a very fruitful endeavor. And what I want to do is tell you a little bit about how we've thought about these problems, how we've approached them, and some of what has been learned. So if we're missing all of these Mendelian traits where there are single mutations with large effect, why haven't we seen them or recognized them as Mendelian traits previously? And in thinking about this, there are a few ways that we imagine this might occur. One example would be there are extremely rare gain-of-function mutations, so that families with these might be exceedingly rare, 1 in several million in the population, and if you've missed it clinically, you may have to wait a long time to see another such family. A second possibility is that there are reasons that we have systematically not been able to recognize that these are single gene disorders. So what if, for example, a disease is caused by a new mutation that is present just in the offspring of unaffected parents, and it has a large effect, and the affected individual is so impaired that they are unlikely to ever pass their genes on to the next generation. In that setting, patients with these diseases will almost always appear sporadically within families, and it will be difficult to recognize that these are caused by single gene mutations. And moreover, the traditional mapping approaches of collecting extended families, following the inheritance of every segment of every chromosome and comparing it to the inheritance of the disease will not get you anywhere. So we think that there are likely systematic problems that have not yet been tackled. We also have recognized that there are extreme phenotypes that arise in the setting of consanguineous union, suggesting that these are potentially recessive disorders where the same mutation has traveled down both branches of the consanguineous loop and has become homozygous in the affected offspring. And then lastly, there are likely diseases that are caused by somatic mutation outside the usual context of cancer, which we have long recognized is a disease largely driven by somatic mutation. So thinking about this has led to strategic approaches that we have taken in our lab, and many other labs around the world have taken similar approaches, and I'll give a few recent examples from our group and others. So one of these starts with a very interesting observation of an unusual phenotype in Iran. And shown on this slide are 3 pedigrees of individuals who have early coronary artery disease, a very unusual form of central obesity, diabetes, and hypertension. And what was striking was that in these families, there's profound central obesity that is so characteristic that physicians and family members can recognize who has this trait and who doesn't within the family. And in one particular region in Iran, 3 such families were identified, and they were not known to be related to one another, but ultimately were shown to share a relationship and have the same mutation driving their disease in each case, which is a mutation in a gene called DYRK1B. And shown on this slide is that these patients have a mutation that is driving unusual development of fat cells far greater than normal cells or cells that are overexpressing the normal version of DYRK1B. This is a gain-of-function mutation in DYRK1B that appears to be at least driving the obesity part of this disorder, and is an example of a founder mutation that is shared by these affected individuals. De novo mutations that I've described previously are another class, and I want to give one dramatic example from our experience at Yale. So the story begins with this individual who was born in an outside hospital and transferred to Yale on day 15 of life with severe watery diarrhea of unknown cause and fever. He was developing coagulopathy, which led to his transfer to Yale. And Mustafa Koka, a member of this society, who I believe has a poster presentation on a different topic at this meeting, called me and said, "This patient is doing poorly, and I'm very concerned that he's not going to survive. He's been seen by virtually every consult service at Yale, and nobody has a good idea as to what the unifying diagnosis is. What do you think about sequencing him to see if we could figure out his cause?" Well, of course, that's a long shot in many cases, but the history was so unusual and distinctive where we thought it was worth pulling out all the stops and giving a try. So in 7 days, we succeeded in getting samples, making DNA, sequencing all the genes in the genome of him and his reportedly healthy parents, analyzed all of the data, and were very disappointed to find that there were no new mutations in the boy that were at all suggestive of causation of his disorder. Unfortunately, he died the day before we completed the analysis from a massive pulmonary hemorrhage. And we were very obviously upset with the outcome. But we were even more upset as we were collecting his medical records from the outside hospital to learn that his reportedly healthy father had that day been hospitalized with acute respiratory distress syndrome, fever, and coagulopathy, and was now intubated in the intensive care unit. This obviously struck us as highly unusual, and we simply thought to look at whether there were any interesting variants that were shared by the father and his deceased son. And interestingly, Neil Romberg, a pediatric allergy and immunology fellow, had seen the child the day before he died and said, "I see signs of systemic inflammation in this infant," and sent off blood samples before he died. It turned out that there was one variant that was shared by the father and the son that had never been seen before, that was in a gene called NLRC4, which is involved in the innate immune system and is normally used to detect bacterial proteins within the cell and trigger an innate immune response, leading to formation of inflammasomes and production of caspase, and ultimately production of interleukin-1 beta and interleukin-18. So interestingly, this mutation was in one of the genes in this innate immune pathway, And both the child and the father proved to have exceedingly high levels of both of these proteins. And moreover, the father's mutation turned out to be a de novo mutation, absent in his parents. So the father ultimately was put on high-dose immunosuppression, and after a 7-week, very rocky course, ultimately recovered. And at that time, we asked him more detail about his medical history. And he said, yeah, I've been healthy all my life. I get fevers now and again. And we asked, what kind of fevers? He said, well, I know that when I incur physical or emotional stress, I'll spike a fever to 104 to 106, and I'll generally get better in a couple of days. And how about your neonatal history? Well, actually, I was hospitalized at Boston Children's Hospital the first month of my life, and in fact, I was written up by my doctor named Dr. Schwachman, who wrote me up as one of the first patients who received total parenteral nutrition because I had intractable diarrhea for the first several months of my life. But I ultimately recovered and have done fine since. Turns out he has a son by a different mother who also has a poorly diagnosed GI problem. When we asked his mother whether he suffers fevers, she said, oh, fevers, let me tell you about fevers. If my son doesn't get enough sleep the night before, I know that the next day he'll start having diarrhea and we'll have a fever to 104 by 6 o'clock, and I'll cool him off and he'll be better in a couple of days. So he also had this mutation. So we showed biochemically that it's a gain-of-function mutation, and this is a nice example where astute clinical observation coupled with this technology both defined a new disease and identified its cause and also suggested a therapy. There are monoclonal antibodies to interleukin-1b that are used therapeutically, and this knocks down the levels of these cytokines, which we think will be a useful preventive medicine in these patients. Another example of de novo mutation that we've worked on is a project, a large project in collaboration with many centers around the United States on congenital heart disease. A frequent congenital abnormality, and the severe forms we think fit the bill for thinking about the role of de novo mutations, because prior to the advent of cardiopulmonary bypass, these children frequently did not survive the first years of life. And so as a consequence, we sequenced a large cohort of about 360 parent-offspring trios where this child had a severe congenital heart defect, but the parents and everyone else in the family was ostensibly healthy or free from congenital heart disease. And we asked, is there an excess frequency of de novo mutation? The answer is no. In cases and controls, the number of new mutations is roughly the same. But it turns out where they land makes all the difference. So if you look at genes that are highly expressed in the developing heart at the key times in development at which these developmental decisions and patterning of the heart are being made, there's a marked excess of de novo mutations in those genes in patients with congenital heart disease compared to their control individuals shown here. Whereas genes that are not expressed in the right time and right place in the developing heart show no difference in the distribution between cases and controls. And moreover, there's one pathway that kind of jumps off the page as being mutated more frequently than expected by chance. And these are genes that modify histone proteins. So the DNA in the chromosome is wrapped around these histone proteins called nucleosomes, and they're modified in order to help turn genes on and turn genes off in development and in other forms of regulation. And fascinatingly, there are mutations that abrogate the production of specific modifications in these proteins, and there are mutations that inhibit the production of the mark, mutations that inhibit the removal of the mark, and mutations in genes that read these marks. And what this suggests is that these are highly dosage-sensitive genes because you need only one defective copy of these genes in order to get a phenotype, and a little bit too little or a little bit too much is enough to drive these phenotypes. And this, we think, is likely playing a role in a significant fraction of congenital heart disease. Overall, we think de novo mutation accounts for about 10% of congenital heart disease of this severe form, and these specific class of mutations are almost half of that total. Now, in parallel, we and others have studied other congenital abnormalities, and the largest of these has been a large study funded by the Simons Foundation and NIH on autism. And now several thousand parent-offspring trios have been studied using this same technology, looking for new mutations. And fascinatingly, again, about 10% of patients with autism prove to have de novo mutations as the driving causation. And in this case, there are two large classes that are driving the disease. One of these are genes involved in synaptic transmission, shown on the left part of the slide, and on the right side of the slide are chromatin-modifying proteins. Again, genes that are driving the modification of these histone proteins that are involved in regulating the turning on and turning off of genes in key developmental times and windows. And at this meeting, I think Bruce Gelb will expand on this discussion. I mentioned rare diseases arising in the setting of consanguineous union, and this has led to development of a genotype-driven approach to genetics. We've traditionally recognized phenotypes, tried very hard to get patients who precisely match one another, and then analyzed those as a distinct group. The sequencing approach suggests instead a genotype-driven approach because many of these patients are simply unclassified. And a nice example of this is congenital brain malformations, where they're highly heterogeneous. Most physicians have never seen more than one of a specific type, and as a consequence, many of these are not well categorized. And as a consequence, my colleague at Yale, Marat Ganel, has collected a very large cohort of these patients with quite stunning cortical abnormalities in cortical brain development, and has simply started sequencing large cohorts of these individuals. And then from identifying rare patients with particularly compelling mutations suggesting homozygous loss of function mutation, has gone back and pattern matched and said, can I find other patients who look like have the brain malformation like this first patient, and have sequenced those additional patients, and has identified a large number of genes that, when mutated, cause congenital malformations of the brain. And here are three examples from his work. But the brainstem is completely unremarkable. Here's another defect caused by, and these are caused by mutations in a gene called WDR62. Mutations in a laminin C3 cause a selective loss of cortical folds in the occipital cortex and a characteristic seizure disorder. And mutations in CLIP1 found by Marat and Joe Gleason, and also Jim Lupsky's lab at Baylor, cause another characteristic malformation of the brain. A pediatric nephrologist in my laboratory, Matthew Lemaire, who is now at Toronto Children's Hospital, used this same approach to study hemolytic uremic syndrome and identified a novel form of atypical hemolytic uremic syndrome that is caused by mutations in diacylglycerol kinase. And identified a number of families with a characteristic presentation, presenting in the first year of life with hemolysis, thrombocytopenia, and kidney failure. These patients, unlike those with mutations in the complement cascade, do not respond to anti-complement treatment, and they can be cured by renal transplantation, again, unlike most patients with atypical HUS due to mutations in the complement cascade. We believe the reason that these patients have their thrombotic diathesis is that diacylglycerol kinase epsilon is involved in metabolizing diacylglycerol, which is an active signaling molecule, to the inactive phosphatidic acid. And in the absence of this gene, they have persistent diacylglycerol signaling, leading to activation of protein kinase C, which is a prothrombotic molecule in the professional thrombotic cells of platelets and endothelial cells. A recent example that is a nice example of gene by environment interaction comes from work of Bridget Stewart, who's a pediatrician at UT Southwestern, working with Christine Garcia. And the approach we took here was to take 100 unrelated patients with idiopathic pulmonary fibrosis and simply sequence them and look to see whether we could find an excess burden of mutations in particular genes that might be driving this disease. And then if we do find those, go back in the individual families and look at the segregation of these variants and compare that to the segregation of idiopathic pulmonary fibrosis or other lung disease. And so in the analysis, there were 2 genes that were mutated far more often than expected by chance. And these were genes called PARN and ARTEL1. And neither of these had previously been implicated in idiopathic pulmonary fibrosis. These mutations in families precisely co-segregated. Every affected individual with idiopathic pulmonary fibrosis within a family shared the same mutation, something that was very unlikely to occur by chance. There were also individuals with less well-defined lung disease who shared the mutation, and there were also individuals who did not harbor the mutation, who did not have the disease but had the mutation. Nice examples of incomplete penetrance. And in fact, these families are highly loaded for individuals with environmental exposures. For example, welders, individuals who raise farm animals were very overrepresented in these families, and within these families, these individuals who had these occupations were more likely to present with the disease. And they also had short telomeres, like seen with other mutations in the telomerase subunits that are known to cause idiopathic pulmonary fibrosis. So it's a nice example where there's a gene by environment interaction leading to recognition of a gene that, of new genes that drive this disease that had escaped identification by traditional methods. I'll come back briefly to our love of hypertension, where there are a number of puzzles that we had not been able to solve in the prior era of Mendelian genetics. So back in 2001, we identified a novel family of serine-threonine kinases that, when mutated, caused a novel phenotype of hypertension with hyperkalemia. And at that time, we had no idea what the normal function of these Wnt kinases were, nor how their mutation led to these characteristic disease phenotypes. And over the years, we were successful in showing that these are involved in the regulation of electrolyte flux and intrinsically the balance between salt reabsorption and potassium secretion. But these only explain 15% of patients with these diseases. And with this exome technology, we fairly quickly were able to show that all the rest of the patients had mutations in one of 2 genes, a gene called CULLIN3, and all of the mutations were de novo and caused skipping of 1 exon in the gene. And the other mutations were in a gene called Kelch-like-3. And CULLIN3 and Kelch-like-3 turn out to be components of a ubiquitin ligase that binds to target molecules and targets these molecules for degradation following their ubiquitination. And again, at the time, we knew nothing about what these molecules are normally targeting, nor how the mutations would act. And a very talented MD PhD fellow in the laboratory, Shigeru Shibata, has put all of this together and shown that, in fact, the normal targets for Kelch-like-3 are the Wnt kinases, the other genes that are mutated in this disease, and that the mutations that cause disease prevent the interaction of this targeting molecule with Wnt4, and the mutations in the Wnt kinases prevent this interaction as well. And moreover, this overall pathway is regulated by angiotensin II signaling, and that the key difference in how the kidney sees aldosterone depends on whether there's angiotensin II around or not. In the presence of angiotensin II, the Wnt kinases drive a pathway that lead to maximize salt reabsorption and diminish K secretion, and in the absence of angiotensin II, the kinases do the opposite. So this has important implications clinically. We've known for a long time that if we give humans increased dietary potassium, that lowers blood pressure, but we haven't known anything about the mechanism. It enters into the same final common pathway regulating how the kidney handles salt. Lastly, I'll mention another mystery that gets to somatic mutation in disease, and this is aldosterone-producing adenomas. These are tumors that are found in 5 to 10% of severely hypertensive patients around the world. These are curiously benign tumors. They rarely invade or metastasize, and we wondered whether there might in fact be simple biological mechanisms that link the constitutive cell proliferation and hormone production by these tumors. And it turns out that that is in fact the case, that these tumors are characteristically caused by single gene mutations. The first of these are mutations in a potassium channel called KCNJ5, and two specific mutations account for more than 40% of these tumors worldwide. And these mutations alter the selectivity filter of this channel so that rather than simply conducting potassium out, they can also conduct sodium into the cell, and the selectivity ratio for potassium over sodium goes from being extremely high in favor of sodium to 1:1. So these are wide open to both sodium and potassium. The consequence of this single mutation is that cell depolarization is the normal signal acutely for aldosterone production and chronically for cell proliferation. And so by introducing a sodium permeability into these cells depolarize and activates the production of aldosterone, causes cell proliferation. And the proof that these mutations, single mutations, are sufficient to produce these tumors comes from the recognition of rare patients who have these identical mutations as germline mutations, and in this setting, these patients develop adrenal glands the size of small baseballs in the first 5 years of life, have massive aldosteronism, and ultimately require bilateral adrenalectomy as a consequence of these single gene mutations. We've shown that this same principle applies to other endocrine-producing tumors, Cushing's tumors of the adrenal gland that constitutively make cortisol, and insulin-producing tumors of the beta cells of the pancreas are all driven commonly by single mutations that have large effect by co-opting the normal machinery that drives hormone production and cell proliferation. So, starting over the last 3.5 years, the Centers for Mendelian Genomics have been tackling problems like these, and to date we collectively have sequenced about 18,000 patients from all over the world. Pediatrics, not surprisingly, has been highly overrepresented because of the large number of extreme phenotypes that we recognize clinically in the pediatric population. We've identified more than 375 new genes linked to human traits that through this process, and there are many more in the pipeline. And I would encourage all of you to think about those rare patients who you see in your clinic who you say, I know they've got something, but I don't know what it is. We have the technology now to figure a large fraction of these out. In particular, if you've seen more than one of them, if you've seen 2 unrelated patients who have the same rare phenotype, we can figure out a pretty large fraction of these. But we're not perfect. We've got a long way to go, but I think our goals should be now we have the tools unequivocally to determine the consequence of mutation of every gene in the human genome. These are going to define the therapeutic approaches to these diseases for the next millennia, and we ought to get on with this. We have a much longer way to go to understand the other 99% of the genome, and it's presently unclear how important, how frequently the rest of the genome will play a role in having rare mutations with large effect, and that's something that I think everyone in the field passionately wants to see. We think that there's a clear path forward to finding loss of function mutations in every gene in the genome. If we look at the offspring of first cousins, if we sequence 500 such patients, we find that almost every one of them has a homozygous loss of function mutation in a gene that we have never seen knocked out in the homozygous form before. The challenge here will be to link phenotype to this. If they haven't seen the right environmental provocation, they may be healthy, and so trying to figure out how to proceed from this will remain something of a challenge, but I'm very optimistic that we'll fill out this map in a relatively short period of time. So we think about rolling this into the clinic with diagnostics and therapeutics. There are opportunities and challenges. I think we're well past the point where we doubt that sequencing individual tumors coming out of patients with cancer is going to be rewarding. We are routinely sequencing these patients for at least a handful of actionable mutations, and this is increasing in its utility as we go forward. In Connecticut, we screen every newborn for about 40 diseases with indirect tests. It would be surprising to me if we didn't find that we can do a better job of this by sequencing the genes that underlie these disorders, and there's work going on around the country to test these ideas. Clinical mysteries I've mentioned, and I think it's clear that if every smart physician in a hospital, every consult service can't figure out what the disease is for a relatively trivial amount of money compared to the cost of a stay in an intensive care unit for a day, we can know whether there's a genetic contribution that we can identify. Plasma DNA sequencing for sequencing DNA that is circulating in the blood as a means of prenatal diagnosis is real now and getting better all the time. And as a way of screening for cancer as well as for monitoring response to therapy is becoming a clinical reality. And all of this, of course, drives the question of should we be doing this much more frequently, and should we be applying this to the general population? And the answer to that is we really don't know. We don't know how often we will find anything that is useful to patients, particularly healthy patients. If we sequence healthy 50-year-olds, it's very unlikely that we're going to find things that are going to be medically important that would drive their disease. But we don't know very much about what would be the consequence of sequencing everyone at birth, with the exception that today we can say with certainty that we would find many variants of unknown significance. And so if we sequenced everyone in this room, every one of us would have 30 to 50 variants that change an encoded protein that we have never seen before in the world's experience. And as a consequence, there is, I think, legitimate concern that if our goal is to improve health, we have to make sure that we don't simply increase neurosis in the population about diseases that we are not actually likely to be at risk of. So how we get from where we are today to where we want to be is obviously a challenge, and these are not issues that I alone am interested in. Obviously there's a large field, but we also have a very strong proponent in this field as well, and that's President Obama. So President Obama, in his State of the Union speech this year, proposed a precision medicine initiative, which I have the arduous task of co-chairing the planning for. The goals are to roll out the increased use of genomics in driving decisions in cancer, but also a proposal to establish a 1 million-person cohort and get very deep phenotyping and establish a longitudinal cohort, follow these patients for many years, have complete genomic information, and ultimately try to link genomic information, clinical records, and clinical outcomes with a goal of ultimately identifying new biomarkers that will be predictive of future risk, predictive of specific disease currently, and this is a very ambitious goal that I think we can look forward to. And I think in the pediatric population, key questions are what role will the pediatric population play in this precision medicine initiative, and this is up for discussion, and so I would encourage you to make sure that your societies have robust lines of communication as this goes forward. So I want to close then and thank you very much for your attention. I think the future is bright, but there's an enormous amount of work yet to be done. Thank you so much.
Thank you very much, and that concludes our opening session presentation. Thank you for attending, and I hope you enjoy the rest of the meeting.